Sir, we can start.
Yeah, okay, thank you, sir. Good morning, dear. Good morning, dear all, and welcome to day 3 of 6 days online AICTE-ISTE sponsored refresher program. I would like to take this opportunity to express our hearty welcome our speaker for this session, Dr. Jyutika Rajwade, ma'am. Welcome, ma'am.
Yeah, I'm happy
Ma'am
to
is
be
currently
here.
working. Thank you, ma'am. Ma'am is currently working as scientist at Nanobioscience Group, Agarkar Institute Research Institute, Pune. Ma'am specialization includes metal microbe interactions and their applications, biotechnology, application of nanoscience and nanotechnology in medicine, agriculture, and environment. Ma'am has 40 research papers, 4 book chapters, and accepts accepted in conferences more than 35 and 10 patents to her credit. Now I request Dr. Rajwade ma'am for her talk on nanotechnology based rapid point of care diagnostics. Please ma'am.
Yeah, thank you for those words, and I'm really happy to be here, part of this particular refreshers program, and I'm sure that I can enthuse the audience with some little work that we are doing in the area of nanobioscience. So, with your permission, I will now share screen.
Yeah, please ma'am.
Yeah. Hello.
Yeah, it's visible ma'am.
Yeah, thank you so much. And we will begin the talk today. Our institute is in Pune, and many of the participants might be aware that the institute is located in a very, it's like a educational hub with institutes like Ferguson College and Gokhale Institute by our side. And on one side, we have the Pune University and the NCL. These are the old campuses. Bhandarkar Oriental Research Institute, many people might be knowing that also. And newer institutes like IISER Pune have also come up. Our institute is completing 75 years of its existence, and it was established in 1946. And this is our institute main building. I am at the Nanobioscience group, and today we are going to discuss a little work, our work on nanomaterials based rapid point of care diagnostics. Some I would take questions in between also if there are any doubts, or then we can reserve your questions for the end of the talk. So with not much of more introduction, we will begin. So when we are talking about diagnostics, we will first understand, I think we should first understand the word diagnosis. So the word diagnosis is made up of two parts. The one is dia and the second is gnosi. So dia is to split apart, and the actual meaning is apart, but here it would be split apart, and gnosi is to learn or have knowledge. So thus diagnosis is the identification of the nature of anything, either by process of elimination or any other analytical methods. And this is the importance of the diagnosis, that it is like a computer program. It follows a particular algorithm, and this, many things are eliminated, and sophisticated methods and techniques are these are the analytical methods or analytical tools which are used in diagnosis. An integral part of diagnosis is medical diagnosis, because as pharmacy students you are very close to the medical personnel. So in medical, what is medical diagnosis? So medical diagnosis is the label given to a condition or a disease which is identified by its signs and symptoms and from the results of diagnostic procedures. So you all know that there are several diagnostic tests. They are categorized as hematological test or biochemical test. Then there are a lot of imaging techniques like X-ray, ECG, ultrasound, and newer techniques like MRI, PET, CT are now available in practically every district, town and district at the state levels. So we have a battery of tests which could be classified as diagnostic tests. So the information which is gathered from these diagnostic tests help the physician to diagnose a particular medical condition. So when we have the hematological and biochemical techniques, why each and every technique cannot be classified as a rapid diagnostic test? No, because sometimes these tests, even if it is a hematological test or biochemical test, the results are obtained with in a long the time to result is quite long, which we call it as a turnaround time. So if you want to develop rapid diagnostic tests, then the results of the test should be available in minutes and not in hours. These diagnostics tests have to be accurate, they have to be simple to use, low cost, easy to interpret, and stable under extreme conditions. Further, we require that the sample should have little or no processing. And these rapid diagnostic tests often include the point of care test for the doctors and walk away test, that is the home test, which can be conducted by the person or his accompany himself. So this is the way a rapid, and where these are the characteristics that make rapid diagnostic tests. Now, what rapid diagnostics could test is the question, and ultimately what we want to achieve using the rapid diagnostics. So, being a microbiologist by training, I thought of giving few examples related to microbiology. So, typically, whether the infection is a bacterial infection or a viral infection, this is one kind of a question which can be answered. Then the second is the type of the bacterium. So in a rapid diagnostic, we could have the detection whether this is a gram-positive bacterial infection or a gram-negative bacterial infection, or a classical a classical agent like Mycobacterium tuberculosis. So the bacterial type can be one of the causative agent, that is which type of bacteria are causing the infection. This is the question that is asked. In the third, we could ask a question like determine the resistance of the organism. So for example, the person is suffering from a bacterial infection, and then the antibiotics need to be transferred, need to be given or prescribed to the individual. So it is known that antibiotic resistance is an increase, and all the bacteria are turning resistant to the antibiotics. So which antibiotic to prescribe? So those kind of tests could be included in rapid diagnostics. Similarly, to just as there is resistance, which are the drugs that the bacteria are susceptible to will be the question, and therefore there will be the rapid diagnostic like susceptibility. So if you go on all these, if you take steps 1, 2, 3, 4, you will see that the size of the circles are gradually increasing. This means that each particular test is giving you more and more directed answers because of your questions are directed. And therefore, if you can solve all these questions using rapid methods, then we can definitely have early treatment. The main thing is about rapid diagnostic is to have start early on treatment. Now, what early treatment can happen? So if you see this particular table, which I have taken from a review published on antimicrobial resistance, it shows that diagnostics can reduce the cost for hospital. So if the cost of hospital are reduced, then there is less burden on our healthcare systems and the patients have to lose less money. So if there is proper diagnosis of the disease using proper diagnostics, then the patient and is benefited because he ultimately spends less, and our healthcare systems are also less burdened. And this is what can be achieved with good quality of diagnostics. Then what are these tests? When we are talking about point of care diagnostics or in general diagnostics, so what do they do? So this is the question, and why they matter. For example, there are some tests which are categorized as screening test, some categorized as assessment. This is diagnosis assessment, and some are categorized as management tests to manage the infection. So I have grouped the screening, assessment, and management on the left side. And what does the test do? So let's have a quick understanding on this part also. So the screening helps in risk assessment. So the screening test usually go beyond family and medical history to evaluate the likelihood of an individual developing a particular condition. So risk assessment, particularly in the COVID area, in the COVID pandemic, we have seen that there was lot of screening done, where continue at present also, because of this new variant which has been introduced, that is the Omicron, there is a lot of screening which is done, and this is a part of risk assessment. So here also, in this particular area also, we need to have rapid diagnostics. Then there comes the early detection. So routine and at-risk tests that may catch the disease at early stages. So in in detection, we should have, if the I'll tell you one simple example. Like if we detect the disease at an early stage, the treatment can be started quite early, and if the right treatment is started at the right time, then the patient recovers early. So early detection and early diagnosis is extremely important. Then during the procedure also, there requires an assessment, and diagnosis and staging and prognosis are these type of the tests that give an assessment of the situation. So whether the treatment outcomes are proper or not, whether the treatment needs to be changed, all these examples come under, all these questions rather are answered during the assessment. And then there comes the management, that is very important, and these tests also help in the selection of therapy as well as monitoring and the or treatment assessment. So to ensure ongoing safety and effectiveness of the prescribed treatments or the course of care, this particular thing is important. So this particular part is more in the cancer patients, where the disease has to be really managed, and in the step of monitoring and the treatment assessment has to be done in a continuous manner. So in the right portion of this particular table, I have given the importance of screening, assessment, and management in details. So in general, I would like to sum up by saying that all these tests are important in their own way, and these help where there are certain views, like the doctor has prescribed so many tests, why do I really need them? That is the outlook of the patients, but that should not be there. And the importance of screening, assessment, and management is very much there in to achieve the treatment and disease-free individuals. So then when we come to the diagnostics, then we just have to have a brief background that what is the current diagnostic need in the Indian market. So because we all know that now it's not good to call India as a developing country. We have actually, really speaking, we have crossed that stage, and now we can see that there are several startups, several biotech companies which are coming up. Not only biotech, there are all the enthusiastic young minds who are at work practically in all the aspects, and they are really planning to have different things to the benefit of the society. Our government also is giving quite a lot of importance to the startup groups, and this is going to actually change the landscape of India in general. And therefore diagnostic segment is one of the segment which is going to capture the Indian market or make its way through the Indian market. So to understand what are the diagnostic needs, I have just put in a few points. So the tests should be affordable, user friendly, they should have real clinical value with high specificity and high sensitivity. Then the purchasing decision in the case of the test often depends on the perceived value and the utility. So if the patient can afford it, or a public hospital can afford it, or and they can afford and provide it free of charge on low cost, so this is the purchasing decision, and the value and the utility of the test will decide the purchasing decision. Then we also need multiplex tests, like simultaneously 2 to 3 things or more should be able to detect. So multiplexing is much better than single separate tests. So for example, there is a condition when the person just has high fever. Sometimes he's saying that yes, there is pain in the joints. Sometimes he's having complaining of general pain, but then the antibiotic is not working, so definitely it is kind of a viral infection. So if there is a test which can differentiate between chikungunya, between dengue, between say any other viral like hepatitis, so this kind of test, if they are available, then that could help the doctor in a much better way to give a directed therapy. So multiplex test development of multiplex tests is definitely important. Then we know that our India is a big country and we have extreme weather conditions. So during transport, like for example, something which is made in Madhya Pradesh has to be transported to Tamil Nadu. Then there are a lot of weather condition changes are there, especially during the monsoons, during the summers. And tolerance to extreme weather conditions has to be there, so that when the infrastructure is not all that good, the test can be, or the diagnostic can be transferred, and especially in the rural areas, and it could work. Then the rapid test for diagnosis of communicable diseases as well as neonatal sepsis and pneumonia. So if you consider, there are a lot of hospitalizations for small children who are just neonatals, that is the just born newborn individuals. So these children are the future of India, and they definitely require lot of care. So this particular problems of sepsis and pneumonia so specifically need to be addressed by developing rapid tests. Then one more thing for the Indian market is that our test and the diagnostic should not have any requirements for power or electricity. So sometimes the diagnostic can be battery operated, that is also possible, but high-end equipment should not be really required. And therefore, and the last part which should not neglect is the disposal of the diagnostic kit parts after use, because here now we are developing the tests which are single-use tests in many cases. So this particular single-use cases should be such that after the test is over, then it should not cause pollution in the environment. So the whole picture has to be very clear, and every point has to be addressed very well. So now at present we have several point of care assays. I have listed here, like from tuberculosis to diabetes, cardiovascular diseases. Many of these point of care assays are being developed, and this particular list is definitely going to increase at least few folds in the next few years. Now, here we have met to understand how nanomaterials can be helping us in the diagnostics. So let's have a quick idea about nanotechnology. I'm sure many of the enthusiastic refreshers who have joined this particular course have definitely seen these kind of slides. But I have just included this slide as a transition from what we started with the diagnosis, and now we are moving to nanotechnology. So in this particular slide, I have just shown the size perspective of various materials. The humans are on the rightmost corner of the slide, whereas we have molecules like hemoglobin, which are particularly in the size range of 1 nanometer to even 1 angstrom. So there are proteins, then there are assemblages of proteins like forming the viral particles. Then we have our microorganisms. Then there are red blood cells, which are around 7 or 8 micrometer in the size range. And then we have hair, hand, which I have shown. So here, whenever we are talking about nanotechnology, we are talking about the materials which have one of the dimension as 1 angstrom to 100 nanometers. So this is the size range that we are talking about, and we are discussing nanomaterials mainly from the perspective like the synthesis of materials usually takes down from the top down as well as the bottom up approach. But in general, if you see that wherever there is bulk material, you go on chopping the bulk material and then come to some arrangement which is just an atomic structure. So the atomic structure containing repeated arrangement of unit cells are can be referred to as nanomaterials in general. So where do we see nanomaterials, or are always nanomaterials manufactured is the question. So no, there are several materials which have nanoscale structures. Like for example, milk is a nanoscale colloid. So the if you can see the or if you have an idea about the chemistry of milk, you will understand that there is fat, there is protein, there is solid non-fat, and all these things are extremely well blended to form the colloid of milk. So this is a natural occurring material, and it is definitely it is classified as a nanomaterial. Then we have functional proteins, DNA, RNA, also which are in the nanoscale. And then here I have shown beautiful pictures of these glass houses of the diatoms. So look at the good symmetry and arrangement of all these structures, and these are indeed naturally forming. The diatom is building its own glass house using the proteins and the silica which is available in nature. So it is constructing its own glass house step by step and forming these intricate, well-crafted structures. Similarly, in bacteria also, there are certain bacteria which produce specific surface layer glycoproteins. And this particular image, which is a TEM image, shows the regular arrangement of the proteins on the bacterial surface, which form nanostructures. Apart from this, we in this particular environment, when we are using a lot of chemicals and lot of vehicles, so there are nanoparticles that occur in the vehicle emissions also. There are products of combustion and cooking of food also lead to nanomaterial synthesis. So nanoscale or nanomaterials are not very new, but the term nanotechnology is relatively new, which was coined by in 1974 by Norio Taniguchi. So this is a recent development, but we all know that nanomaterials or nanoparticles have existed. Now, at present, there is a lot of research in the area of nanomaterials, and the question definitely will be asked. So, what are the major categories of nanomaterials? So, I have shown in this particular slide that there are practically all categories of materials. I have included semiconductor materials like zinc sulfide, cadmium sulfide. Then there are antimicrobial nanomaterials made up of silver, zinc oxide, titanium dioxide, copper oxide, etc. Then we have even these biotemplated nanomaterials. Just in the earlier slide, we have discussed them. The S layer, the spore structure of a bacterium also is nothing but proteins arranged in a very typical manner and forming nanoscale structures. And apart from these, there are hybrid, that is the organic, inorganic, or the bimetallic materials, which are also classified as nanomaterials. So we have at least 12 categories of nanomaterials, which are organic, inorganic, and hybrid materials. So this is the general classification. Now, usually when we say that we want to have some rapid diagnostics, then the first question or first thing comes to mind is what are everybody or the literature is there saying that the importance of gold nanoparticles in the assays. So let's understand why gold nanoparticles are important. So in general, for the point of care assays or in various assays in vitro diagnostic applications, gold has a special role to play. And this is because of a particular phenomenon called localized surface plasmon resonance, which is exhibited by the gold nanoparticles. So the nanomaterials themselves in applied electric field, there is a they show this particular phenomenon of LSPR, and this LSPR is responsible for the color of the gold that we can see with our naked eye. The bulk gold which we see everywhere is yellow, bright metallic in color, whereas the gold nanoparticle solutions are typically red in color. So there are size dependent colors also, and you see that 19 to 35 nanometer gold appears usually wine red in color, and as you go on the higher scale, that is 50 or 60 nanometer gold particles usually take a grayish bluish shade, and then you get gold which is blue in color. So these kind of shape and size dependent colors one can see in gold nanoparticles. And the beauty of these particles is that their surface can be functionalized with several molecules. So I have shown here that the surface of the molecule can be functionalized with DNA, with antibodies, or even with different proteins, then targeting ligands. All these uh, 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 can form structures on the gold nanoparticles. And then these, with all these decorated materials, these particles can be used to spot or to tag small. They can be they are already functionalized particles, and they can be used to recognize small molecules, proteins, nucleic acids, even pathogens and cancer cells. So thus gold is a very versatile material, and it can apart from LSPR based diagnostics, several diagnostics based on electrochemical reactions or fluorescence reactions can also be developed dependent on gold. And then there are these point of care devices, which we will see the examples in the few next slides. And here gold material as such, nanoscale gold, if it is deposited, then some SERS, that is surface enhanced Raman spectroscopy devices, also can be developed using these gold nanomaterials. Coming to the actual use of nanotechnology in point of care diagnostics, several field usable diagnostics can be developed using nanotechnology. And here we have these devices which can be used to find out the pathogens, or to detect the pathogens, the biomarkers, the some heavy metals and inorganic contaminants, the pesticides, herbicide residues, etc. All can be detected using nanotechnology. So actually what happens is there are nanomaterials which could be used in diagnostics. They could be nanotubes of say carbon or silver nanoparticles, then magnetic nanoparticles. We will see the examples in the upcoming slide. So the nanoscale structures and materials can be used to form the products with nanoscale features also. So these can be immobilized and developed into some matrices or contrast agents and optical components also. So here the nanomaterials are there, then we could have nano intermediates, and then ultimately we could have nanotechnology enabled tools, which are nothing but nanosensors or microarray, lab on chip, etc. So this would be the final tools which incorporate nanotechnology. So it could look like this kind of a chips, or this kind of sensors, this kind of lateral flow devices, or the microfluidics. So we'll have a brief on all of these. Then when one as developing point of care assays, when we started developing point of care assays in our lab, then any such assay would have a recognition element, and then there could be some visual readout which is required. So we used initially gold nanoparticles as an agent for the visual readout, and then we tried to develop everything in the 3 assay formats. So first assay format was magnetic immunocapture, flow through assay, and lateral flow assay. So those 3 platforms we developed. And then looking at the recognition element, we found that several things could be used. So these could be the inactivated enzymes can be used. Then there is a whole branch several peptides which are produced or which can be obtained by phage display technique can be used as recognition element. Then we have classical antibody, lectins, etc., which can be used to capture the target of interest. So here are some examples which I have given for the inactivated enzymes. Then with phage displayed peptides, we had 12 more peptides which we obtained via biopanning experiments. And here in our experiment, the target was the white spot syndrome virus antigen, that is the VP28 antigen, which was the target against which these peptides were selected. Then we have used materials like chitosan, then anti-galactomannan antibody, then concanavalin A as these are the con A is a typical example of a lectin which we have used to track the bacterial cells and galactomannan, etc. So actual applications of these now we will see in the next slides. So the first application which we developed was to develop rapid diagnostic for aspergillosis. So aspergillosis is a typical fungal infection, and it occurs in immunocompromised patients. That is the patients undergoing therapy for HIV, for example, or cancer or transplant. So these patients are immunocompromised, and they develop these particular aspergillosis infections. And it is known that this Aspergillus fumigatus is the causative agent in 80 to 90% of these infections. So most of the clinical, doctors are unaware of this particular mycotic disease, and there are PCR and CT scan method, but these are definitely not yet standardized, and there are few kits also for the detection of this fungal infection, but these kits have to be imported, and therefore we try to develop an indigenous diagnostic for detecting these infections. So we took the marker as we wanted to develop a serological method, and for this we selected an antibody detection. We thought that detecting antibody will be easy, but then it faces the limitation that there is low level of antibody production, and these antibodies against the aspergillus are present in the healthy individual always, and therefore the sensitivity of this particular assay is quite low. So then we thought that why not detect the antigen, and the antigen could be the aspergillus derived polysaccharide. So the glucan and the galactomannan are produced when the aspergillus is in the body of the infected individuals, and we thought that galactomannan could be a heat stable and a thermosensitive polysaccharide which should be detected. So we developed the diagnostic test for detection of galactomannan. And here is the test. This was a flow-through assay which was developed, and we proved that the assay works with Aspergillus fumigatus. These are the patient isolates. Then these are the standard cultures of from two culture collections, as well as this is the patient isolate. So we found that these gold nanoparticles based assays could be very useful, and the positive assay could be developed as a red colored drop, which is visible to the naked eye. So sophisticated equipment are not required. Then coming to another infection. So another infection is typically the white spot disease. And this disease is not in humans, but it is in animals. And we all know that currently seafood is in great demand, and seafood is a source of good quality protein, and therefore as part of biosecurity and all these things, to meet the needs of the food, the fish derived materials, or the food that is produced from the sea is an important source of protein. So and India has a very vast coastline. So many farmers are engaged in the aquaculture activities. They carry out activities in the ocean also, and in captivity, that is in the rivers or the lakes also. And one such disease which affects these shrimp is the white spot syndrome virus, which causes the white spot disease. So this disease has this disease spreads very rapidly and result into mass mortality. And here we can observe that these shrimp are, the body of the shrimp develops white patches. So when the person goes to buy the shrimp, then what happens is there is lot of rejection, and the shrimp farmers have to have lot of losses. So then this is not going to be, so there is a need to address this particular problem. And for the diagnostic of WSSV, currently there is this polymerase chain reaction, and the polymerase chain reaction is a molecular method. It's quite sensitive, but it is time consuming. Then it requires skilled personnel. The equipment, that is the PCR machine, is also quite expensive, and it cannot be used on a particular farm. So we need to develop a good diagnostic which will help us for early detection and monitoring of the pathogens. So we decided to develop this particular lateral flow assay based diagnostic, because the farmer is happy if he is just knowing that the result is positive or negative. Only that, yes or no, this kind of result is required. Now, where is this particular assay used? So this particular assay, that is based on the LFA technology, that is the lateral flow assay technology, can be used in medical diagnostic, therapeutic monitoring, agriculture, food safety, and many applications are there. So our lab developed this particular assay using the polyclonal antibody based, this is a polyclonal antibody which was raised in rabbits against this particular antigen, that is the VP28 antigen. And here, this is the workflow. So the gill is collected, it is crushed, and it is used to develop the assay. So the results are obtained within 15 minutes. You can see the line, red line, which is formed, which is indicating the reaction. Here, there are two lines which are formed, which is characteristic of the WSSV. So if WSSV is there, it is getting two lines, and if WSSV is not there, then we get only one line. So there is a control which is incorporated in the assay itself. So positive control is there in the assay. So it shows no cross reactivity also, and results are obtained within 15 minutes. There is no need of any specialized equipment. So this assay is highly reproducible and accurate, and therefore this was developed. Then another assay which we developed for this kind of pathogen itself is the for the detection of mRNA, that is the Macrobacterium rosenbergi nodavirus. And this was a recent publication from our lab, where we optimized the assay using a particular statistical design. And the work on infectious myonecrosis nodavirus infections is going on, and soon we are going to develop this particular assay also. So in the earlier part, I had stressed about the need for multiplexed assays, and here, since we are developing three assays together, then we could add on few more detection agents, that is viruses and bacteria, and definitely can go for multiplexed assays also. So finally, using using all these assays, so we found that the end user has a lot of benefit, and then the purpose of detection of pathogens in aquaculture is solved, because a lot of surveillance and documentation can be done. These assays can contribute to eradication of infections, classification, typing of pathogens, all is possible. And diagnosis at the population level is also possible, or at the individual level. So that can be done easily using these kind of diagnostics. So there are a lot of customer benefits also, and the testing is possible at any stage without dependency on the laboratory. And so these kind of assays can definitely be popular amongst the industry. Then, just as we have discussed all this time about the gold nanoparticles, there could be other nanomaterials also which could be used in lateral flow diagnostics. For example, the carbon nanoparticles can be used. These carbon nanoparticles appear black in color, and they can be functionalized using the antibodies and used for the detection in the lateral flow assay. Similarly, selenium nanoparticles. These nanoparticles are red in color. Selenium 0 valent is red in color, and therefore it can be used as a label where the eye can detect the presence of red color very easily. Similarly, iron oxide nanoparticles can also be used. But with these particles like the iron oxide and the quantum dots, one cannot rely on the eye. We require some few specialized equipment to measure, because with the iron oxide we are studying the magnetic behavior of the material. So to calculate the magnetic strength in that particular area, we need a special equipment. Similarly, to see the quantum dots, we have to flash a source of laser, blue laser, and then these particles will show up as green or red in color, depending on the size. So a few assays for increased sensitivity can rely on the use of iron oxide and the quantum dots as nanomaterials. Then interesting assays can be developed using like the assays which are shown in this particular slide. So they are magnetic viral nanosensors, where these particles, the super paramagnetic particles, are coated with the antibodies, and they can detect the virus. So the virus will be attached to these particles, and they will form a agglomerate or a structure which is nothing but a nano assembly. So because the viral proteins and the antibodies will show complementarity of it, they will form these kind of nano assemblies. So these structures are also these kind of materials are also being developed. Then I will just, in brief, I will discuss some work which is related to nanotechnology and which was carried out in our lab few years ago. And this was a particular case where a diagnostic for rapid detection of antimicrobial susceptibility was developed. And we know that urinary tract infections are one of the commonest nosocomial infections in the, so they are hospital acquired infections, and the patients who are admitted to hospitals or are advised stay in the hospital develop these infections. So they are very popular reports in India, second only to respiratory infections and other bacterial infections also. And there is a high incidence of these infections in women, children, and elderly individuals. So, and another thing is this UTI is an important reservoir for selection and transmission of multi-drug resistant bacteria. So we are all worried about this MDR problem, and with the proper choice of antibiotics, this can definitely be curbed. And that is why some work we initiated on these lines also. So this is a typical picture where what is done in empirical diagnosis, traditional diagnosis, and what a rapid diagnostic test could do. So a rapid diagnostic use could result in optimal treatment very quickly. So therefore, it would reduce the unnecessary prescriptions, and broad spectrum antibiotics can be avoided, and targeted antimicrobial therapy can be given. So therefore, the rapid diagnostic are important. And then I have just shown the way in which the antimicrobial susceptibility testing is done in a conventional way, where say, like for example, this is a urine sample, then it undergoes enrichment and isolation, then a single colony is there, and then we have the antibiogram, or in this particular format or in the well format. And where the culture doesn't grow, we call that this particular drug can be prescribed. Similarly here, the one with the more zone of inhibition can be prescribed. So this is the conventional way. And what we have done is, this is the conventional way which I have put on the left side. On the right side is our procedure, where we take a midstream urine sample, then go for magnetic recovery. We have developed special type of magnetic nanoparticles which can capture the bacteria from the urine, and then we have devised a test where simultaneous antibiotic sensitivity and culture identification is possible. So this is how is the workflow. This is the capture protocol. Then there is a brief step of incubation here. Then we have a plate which is loaded with the substrate, and then we have a visual readout. So this is how the kit with its basic thing looks like. So these are all the different assays, and here we can check which culture is being present in the sample. So we provide these nanoparticles and we provide this medium for enrichment. This is the general diagram, and we conducted a preliminary study with 100 samples, and which were pus cell positive and require antibiotic susceptibility testing. So we contacted a nearby diagnostic lab and got the samples from them, and we analyzed this with respect to the disk diffusion assay and our assay. So we found that there is a fair concordance, because the reading was 0.347, which is the kappa coefficient, which means that both the techniques are fairly concordant, and negative predictive value was 1, which means that if the culture, if our test is negative, then definitely the disk diffusion assay is also going to be negative. So that was the important finding from our work. Then our lab is also engaged in using these microfluidic platforms. They are developing these microfluidic chips in polydimethylsiloxane, which is a polymer. And here it works on these three zones are there. So individual line contains these three zones. So one is the pre-capture zone, which is then that there is a capture zone and there is a detector zone. So initially the microorganism is captured, the rest material moves out, then the magnetic force from this part is released and this magnet is activated. So the material can be washed, whatever one more additional step of washing can be done. Then in this particular step, we can add the fluorescent dye or the the dye is conjugated to the antibodies, and here we can have these bacterial cells and trapped with the antibodies, and this will be the detection zone. So in the detection zone, so this is how the assay would work, and this we proved that quantum dots can be used for capture and detection of these staphylococcus cells. Then we tried to go for a multiplex detection also, where E. coli and Salmonella were detected, and the specificity of the assay was more than 90%. Then you have seen that all these assays, sometimes for particular PCR reactions or any other real-time PCR reactions, we require bacterial DNA to be isolated. And once the DNA is isolated, then we can go for development of the diagnostic. So can nanotechnology be used in DNA diagnosis, DNA isolation also? So we tried to use magnetic nanoparticles themselves, and if we go for classical kits, what a DNA isolation procedure does is that first there is cell lysis, then there is solubilization of DNA, removal of contaminating macromolecules, proteins, lipids, etc. And then in any protocol, we reprecipitate the DNA. So this is the four-step procedure for any DNA isolation protocol. And whatever kits are there in the market, they are based on molecular exclusion, reversed phase chromatography, hydrophobic interaction, or anion exchange chromatography. So with all these particular techniques, they can be used. But there are some drawbacks, that is time consuming, then there requires centrifuge many a times, many steps of washing are there, and then adsorbents, everything are required. And if you consider this particular diagram, if you choose simple methods for diagnosis or isolation of DNA, they give less pure DNA, and the time required is less, but the quality of DNA is also less. So if we go for good methods like density gradient centrifugation, then the time required is also more, but the purity is highest grade. So, but this we cannot do in the lab every day. So we want some method which will give us pure DNA in less time. So what we did is we developed this DNA wrap. We have these surface modified magnetic nanoparticles. We mix them with the bacterial pellet and go only use a brief vortexing on it. Sometimes if vortex is not available, then you can shake it rapidly with the hand also. And then just go for a magnetic separation, elute the DNA with the buffer, and this is our result. So if we go for E. coli cells, then if we treat them with the reagent and capture them, what we see is these cells literally burst open because of these magnetic nanoparticles. The particles are covered with a material which will absorb the DNA also, and we can have the pure DNA eluted. So we observed this on the gel electrophoresis, where PCR products can be imaged, or even the whole cells can be imaged in various dilutions. So very low quantity of cells, even if you have, then you can have the amplification reaction possible, or you can detect them on the gel. So this is how we tried even for the mycobacterium samples, because mycobacterium tuberculosis, mycobacterial cell walls are very tough and they cannot be damaged very easily. And we tried to do this particular reaction using the mycobacterium smegmatis cells, and we found that these cells are also disrupted. So the reagent works with the gram-positive, gram-negative bacteria and difficult to lyse cells. It is generally low on the requirement of reagents, and transportation cost, the storage cost can be reduced because this particular assay or the kit which is developed is stable at room temperature. So these were our findings. And currently we are into paper-based microanalytical devices. So we want something like this. You have seen that our lab has already developed microfluidic platforms, paper platforms in the form of lateral flow assay. Now what we are interested in doing these kind of assays where simultaneously many analytes can be detected using the color or different combinations of gold and silver nanoparticles and other reagents and electrochemical devices also. So this is our aim in for the coming, for the days to come. And in general, we see that the global market, if you just see the global market forecast, in especially for the in vitro diagnostic segment, is that there is a need for all these kind of assays. So molecular assays, histology. Histology is particularly true because the number of cancer cases are rising, and rapid diagnosis of cancer is also very important. Similarly, in infectious microbiology, virology, all in this pandemic, everybody has experienced that these particular assays are really going to be very important. So we need to have automation, and with the molecular platforms for disease testing, we are definitely going to have developed platforms which are cost competitive, alternative, and easy to operate. So this with this I end my talk, and I would take any questions if you have.
Thank you so much, ma'am, for your sharing the research work of our lab with us. I request participants to unmute themselves and ask your question. Before that, I would like to read out one question which is posted in the chat box by respected Dr. Vandana Patrawe, ma'am. Ma'am, should I read out a question?
Yeah, you can read it out.
Could you kindly elaborate on optimization techniques used for such kits? For quantitative determination, how number of nanoparticles reacting with either antigen or antibodies is decided, and how do you know quantitatively how much of nanoparticles are on the control line?
Yeah, thank you, madam. I hope you remember me. We have met in one of our students' Viva. So it was nice that, yeah.
Definitely, I remember.
Yeah. I'll answer your question, ma'am. Actually, while developing the lateral flow diagnostic, we go by the titration protocol, where once we have the nano tracking analyzer with us, so we know the concentration of the nanoparticles which we are adding. We know the optical density of the solution also. So based on the OD reading, and then we react with an excess of antibody to begin with. And this particular thing is actually a very standard titration protocol where we react same number of nanoparticles with varying concentration of the antibody. And then subsequently, after a 30-minute incubation period, we add sodium chloride to it. So whenever there is optimum binding of the antibody to the gold nanoparticles, there will be no aggregation if we add NaCl. So no, we usually decide the antibody to be added on the basis that of that particular concentration, beyond which there is no aggregation. So this is how we choose this particular concentration of the antibody. And then what we do is we collect the antibody and the gold nanoparticles together. So we actually go for a centrifugation reaction, where this particular conjugate is separated, and on the basis of the optical density of the conjugate produced, we add some more of stabilizing agent in the form of a protein like a BSA, or sometimes even glycerin, glycerol can be added. So we add these kind of materials and then again record on the basis of the optical density. We are going for loading on the pad or preparation for the strip. So this is the optimization protocol. And what we have done is, our recent paper is, we have used the Taguchi design of experiments to optimize this entire protocol. So the nanoparticles concentration, the load of the buffer, the selection of the buffer, the quantity of the reagent going on the control line, the test line, all those can be optimized using statistical methods. That is what we have done. I hope
Thank
I've
you, madam.
answered your questions. Yeah.
Yes, yes, definitely. I asked in interest of the crowd who is attending. So very,
Yes,
very
yes.
excellent, I would say, presentation as well as the work which you are doing
Yeah.
in your institute. I'm aware of it. And
Yeah.
I think you should interact much more with pharmaceutical crowd and take this particular technique, for many, many common diseases also, which are still undetected, because of which there is a danger to mankind. In fact, I
Yes,
hear now
yes.
that leprosy is again on rise, and we do not have a rapid kit.
Yeah, yeah, yeah. Actually, the problem is that these are now coming up as emerging and re-emerging diseases. So we have many diseases which were, we forgot that yes, they were there in the population, but now somehow they are coming up again. So we'll definitely take your suggestion on that. Yeah,
Thank you, ma'am. I
yeah,
request participants to unmute themselves and ask if they have any queries.
Dr. Monica have raised the hand.
Yes,
Dr.
Monica, ma'am.
Monica, you can unmute and ask. Dr. Monica Maheshwari.
So I guess there is no question from other delegates. So thank you, delegates, for your queries and Rajwade ma'am for answering them
yeah,
aptly. I
yeah.
would like to take this opportunity to express my sincere thanks to our speaker, Dr. Jyutika Rajwade ma'am. We truly appreciate your dedication for sharing your valuable knowledge and research work with us. Let me summarize your topic. Ma'am has started her presentation with her basics of diagnosis, then meaning of the rapid diagnostic kits, benefits of the rapid diagnosis also. Ma'am has highlighted on importance of the screening, assessment and management. Different kits, kinds of the test available or need to be available in the future and scenario of the diagnostic needs of the Indian market. We came to know about how gold nanoparticles have advantage in diagnostic assays. Thank you for sharing information regarding field usable assay development of detection for white spot syndrome disease developed by your lab and other diagnostic kits too, such as the UTRAP, DNA RAP method for rapid DNA isolation, as well as paper-based microanalytical devices. We wish all the best for your future endeavor. Thank you so much, ma'am.
Yeah, thank you. And my email is there with Dr. D'Souza, and if there are any questions, I would take them on the email also. So
Yes, thank you,
it
ma'am.
was nice interaction. Yeah.
We would like to take this opportunity to felicitate our guest speaker with an appreciation certificate. May I request Dr. John, sir, to please do the honor. Please, sir.
Yeah, thank you, sir.
Thank you so much.
Thank you.
Thank you, sir. Thank you, ma'am.
Yeah.
So now I request all the participants to put on your video mode on for a snapshot with our guest speaker. Sawan sir, please.
Yes, sir.
Is it done, sir?
Wait for 1 minute, sir. Done, sir.
Thank you
Thank
everyone.
you. Yeah,
Thank you once again, ma'am.
Yeah, thank you.
Have a nice day.
Yeah, I'll leave the meeting now.
Yeah, please.
Yeah. Yeah.
There is one direction for all the delegates. The next session, we are beginning with the next session after 1 hour, that is sharp at 12:45. Okay, so please join the session after 1 hour. So we are beginning with our session at 12:45 for the next session. So it will be from 12:45 to 1:45, and the next session will be from 2:30 to 4.